Okay, everybody, it is time to try to finish up chapter 19. And since we had lab, some of this stuff I'm going to try to breeze through when we get to the white blood cells. So, again, this is our objective. This is kind of what we did in lab. And again, in the first video, I talked about how whole blood is divided into our plasma versus our formed elements. And then what we do when we subdivide that plasma, we get plasma proteins are going to be the most abundant solute in the water of the blood. And the most abundant of the plasma proteins, of course, is albumin. And the reason we have albumin, which is made in the liver, is mainly as a fixed solute. So when we're having blood pass through capillaries and leak out, the water osmosis pressure will pull some of that water back into the blood supply, so that way our blood doesn't become more viscous with every time it passes through capillaries. And albumin helps with that. So if the liver was to overproduce albumin, the consequence of that might be extremely high blood pressure, because you would not have, you'd have more osmotic forces holding water in your vessels and less of it leaking out. On the flip side, if you don't make enough albumin, it might be that the extracellular fluid is higher than normal, and so therefore you have edema in your arms and legs and your tissues, and you're not able to then maybe hold the blood pressure as well in your vessels. The next big category we talked about was the globulins, the transport proteins. And again, transport proteins, we're going to have those for things like thyroid hormones, because thyroids are not water soluble. So thyroid binding protein. We're going to have some for some atoms like iron, copper, zinc. So transferrin is an example for iron. And then we're going to have the ones for the steroid hormones like testosterone binding protein. Most of, again, our binding proteins can be made in the liver, but our immunoglobulins are going to be made by our B cells of our immune system as responses to pathogens occur. And they're going to be a large amount of proteins in the globulin category, but they're circulating with the intent to find anything expressing a antigen that would be identified as not self. The next big category of proteins is fibrinogen, and I normally throw in the clotting factors here. Most of those are made by the liver, and again, they are there ready to prevent injury or lossage of blood, try to stymie that lossage of blood if an injury occurs. And so when you, again, clot, you then get left with no clotting factors, no fibrinogen in your plasma of that sample. Let's say if you pulled out blood and put it in a tube and allowed it to clot. So after you spin that blood, the clot will form and fall down with the formed elements on the heavy side, and so you're left with serum. And I do expect you to understand the difference between serum and plasma. And then the last amount of hormones are all the other stuff. So all of the hormones like insulin, FSH, LH, TSH, so thyroid stimulating hormone, and then just a bunch of other little proteins that might be traveling, hitching a ride in your bloodstream. For other solutes other than albumin that are going to have a role in the osmotic rate gradients of our blood pressure in our vessels versus our interstitial fluid versus our intracellular water pressure inside the cells. Those are going to be inorganic molecules such as the electrolytes. So all of those sodium, potassium chloride you hear, we think about when we think about membrane potentials. And then we're going to have a lot of organic solutes here, because all of the small carbohydrates we take in with our gut or move around to provide fuel, such as glucose, fructose, even lactic acid falls into this category, and other small carbohydrates. And then our amino acids, which are moving around to cells. So as they're uptake, they can be made into proteins. And then a lot of organic waste will fall in here. So bilirubin, which we're going to make once we break down red blood cells, urea, ketone acids if we're starving ourselves. All of those acids will be hanging out in the as an organic substrate solute and flowing through our plasma. Okay, so I just wanted to hit those again. All right. So we now need to start focusing our attention on the formed elements, specifically the red blood cells. And we learned in lab that because the red blood cells are 99.9% of your formed elements, that we tend to take a little leap of faith that if we have a decent amount of formed elements, our hematocrit, that percentage of the formed elements that represents oxygen carrying capacity, we have a sufficient amount of red blood cells. So if we want to try to really kind of put the numbers out, how many actual red blood cells do we have in our body? We know we have somewhere around 4 to 6 liters of blood in each person. So 4 to 6 liters, all right? And most of us don't think of that. So, you know, just think 4 gallons of milk, right? 4 milk jugs. How many red blood cells are actually in that blood of our body, okay? So if we take a microliter, now, a microliter, I tell you, is like a really, really, really small amount between your 2 fingers, okay? Typically speaking, males are going to have about 4.5 to 6.3 million red blood cells in that little microliter. Women are going to have a little less, but they still have pretty much 4.2 to 5.5 million. So you have about 5 million blood cells in a small little droplet of blood. So when we looked at those slides under the microscope, let's say there were 3 microliters of blood used to make that slide. If we go with there's 5 million red blood cells per microliter, and we put 3 microliters of cells of blood on the slide, that means that each of those cells should have had about 15 million red blood cells. And we were looking at 15 million red blood cells to try to find the white blood cells that were intermixed, you know, at the 0.1% level. All right. Platelets and white blood cells are going to be, again, that less than 1% of the formed elements. Platelets are important in the clotting process, and they are cell fragments. So kind of like red blood cells, they're not alive anymore. They have a lifespan. They're not going to reproduce, and they're going to circulate and either get used or they'll get reuptaked and recycled at the liver. The white blood cells, the leukocytes, are coming from the bone marrow. Many of them are hitching a ride in our blood to be able to head to lymphatic and immune type tissues. So they're heading to the dermis of your skin. They're heading to the linings of your nose and your mouth. So if something gets past your mucous membrane, your epithelial linings, they're ready to fight that. They're heading to the lining in your stomach, the lining around your reproductive organs, your urethra. They're heading to your lymph nodes, so those things you palpate under your neck and your armpits and your groin area. They're heading to all of those locations where they then take up residence there and then play a role in the immune system. Okay, a few of them will circulate a little longer in the bloodstream, but the majority of them are not trying to hang out for any long period of time in the bloodstream. Okay, now in order to be able to get 5 million red blood cells into a microliter of blood, I need the red blood cell structure to be maximized as possible. So this is where the importance of the red blood cell being a small cell becomes very important. And it needs to not only be a small cell, but it needs to be a small cell that is very stackable and very easy to put a lot of them in a small amount of space without damaging and hurting and breaking apart those cells. All right. Now, as I get to be a small cell, I'm going to limit the amount of volume I have inside of me. So one of the other things I'm going to do with these red blood cells is I am going to have to make some tough choices about what I keep inside the red blood cell for the 100 and so days that it's going to live beyond maturity. All right. And so one of the things I'm going to get rid of is almost every organelle and the nucleus. So red blood cells, by becoming small and therefore maximizing their oxygen carrying capacity, are going to give up the ability to go through mitosis and divide, give up the ability to respond to factors, stresses, and are going to basically have very low energy demands and very low energy making capacity. The advantage to all this is by getting rid of all that stuff out of the cytoplasm, which is a little bit of water, maybe a little bit of some cytoskeletal proteins, has now a lot of space to be packed full of hemoglobin proteins, which is what the red blood cell wants, so it can carry as much oxygen as possible, It does keep a little bit of glucose, and the enzymes and everything that's necessary to turn glucose by glycolysis, which is anaerobic respiration in the cytoplasm, to pyruvate. And then it'll keep some other few little critical enzymes and proteins, such as carbonic anhydrase, which we'll learn about in carbon dioxide carrying possibilities. Okay. Now, because the cell cannot divide, the cell is on a fixed life cycle. So, in your circulating blood, you have mature red blood cells being added to the system every day, and then you have those mature red blood cells live about 120 days and then get cycled out. So every day we're adding a few thousand cells and taking out a few thousand cells, right? And we have to do this because the cells can't live forever. They don't have an infinite source of glucose inside of them. They don't have any mitochondria, DNA, or ability to make more proteins if they get too damaged or too messed up. So this gives us a way to have a cell that's very efficient at carrying oxygen, but it also gives us a cell that has to be cycled in and cycled out constantly. Okay? And the cells that get cycled in, they get added to the system every day, are immature red blood cells. So in this picture here, I pointed out is that they're called reticulocytes, and they're not quite mature red blood cells because they still, as you can see with the purple here, they retain a little bit of like some ribosomes and some RNA and some transfer RNA, and they have a little bit of the machinery still kind of finishing up and working to make more hemoglobin and other critical proteins that are going to be a part of the mature red blood cell cytoplasm. All right. Once reticulocytes come into circulation, we know that it's probably going to be if maybe a day or so, and then they'll like phase into becoming mature red blood cells. And because they go into circulation, and we can kind of expect like a small percentage of red blood cells to be reticulocytes, by measuring the reticulocytes, that is how you can see if you're adding red blood cells with treatment to anemia versus not adding red blood cells. So if normally you add, let's say, 1,000 reticulocytes to your blood every day, if you start measuring someone who's on iron supplements and they're now making 1,500 reticulocytes and adding them to the bloodstream a day, you know that you've upregulated red blood cell production, and now you're adding more red blood cells, and over a few days, those extra reticulocytes, extra red blood cells, will start to accumulate in your red blood cells per microliter, slowly going up, and your total red blood cell count slowly coming up. Okay. All right, so let's go into a mature red blood cell structure a little bit more. So it doesn't have any nucleus, it doesn't have any mitochondria, it doesn't have any organelles, so you don't see anything except the red, and then inside, you know that the red's coming from a large portion of the cytoplasm being full of hemoglobin. All right. Now, the little donut shape is called biconcave, and the advantage to that is when we put them together like plates, they stack, and you can jam pack a large number of these cells in a small amount of volume, like a droplet of blood. All right. So that's how you can get 5 million in a teardrop. Right. The stacking goes back. It's called rouleau or roulette, and that's again what someone named the stacked red blood cells flowing one at a time through the capillaries. Okay. Now, the other advantage of being small with a small volume is that any protein, so I'm going to try to draw, like, let's say hemoglobin right here. This hemoglobin molecule, which is kind of in the center of the cytoplasm over here, technically is still very, very close, a short distance away from the surface of the cell, from this large surface area that the cell has. And the advantage to that is the hemoglobin here versus hemoglobin, let's say right here, when they pass through a capillary, the purple hemoglobin being right next to the membrane, yeah, it's easy to put oxygen on or pull oxygen off. But even though this blue hemoglobin is in the center of the cytoplasm, because it's still, in relative terms, pretty close to the membrane wall of the cell, oxygen getting onloaded or offloaded is still very easily able to bind to this hemoglobin molecule, stay attached, or disassociate. And the advantage to that then gives us that red blood cells have a very large surface area to their very small volume, and that ratio of large to small is a huge advantage on how every cell, when passing through a capillary and passing through that capillary maybe in a fraction of a second, is still able to grab oxygen in the lungs in that small bit of time and unload to 98% saturation, versus in tissues, offload oxygen as necessary to meet tissue demands. Okay. Again, biconcave disc allows stacking. That's a great advantage, so I can get a large number in a small area. And this donut shape does have some flexibility. So the red blood cells, in the event they get turned around, squished a little bit, they're not going to immediately, break open and break apart. Okay. Now, again, hemoglobin is our key protein. Hemoglobin has 2 alpha protein chains and 2 beta protein chains. So in making hemoglobin, I actually transcribe a message RNA, all right? And that message RNA maybe will encode the amino acid, structure for the alpha chain. And then I'll make a second, message RNA chain. And that message RNA chain will be translated into an amino acid, protein that is the beta chain. And then because of the amino acids and their R groups, this is just biochemistry, that chain is going to form twists and twirls that make it these 4 little entities. And then we can stick in the little pocket area a heme molecule. Okay. Then we can put those 4 molecules together, and that 4 molecule bound to each other, coexisting as one kind of entity, is our hemoglobin protein. Okay, and we're going to put all these proteins, as many as possible, into our cytoplasm of our red blood cells. So one hemoglobin protein has 4 hemes in each of its chains, and that heme contains an iron core. That's where our oxygen gas wants to bind. All right, and so our oxygen gas wants to bind to each of those hemoglobins. Now, if a hemoglobin doesn't have any oxygen, the first one to get on is going to be the most difficult. But conversely, if it's the most difficult to attach, it's the hardest to pull off. All right, so the binding of oxygen to the first heme is hard, so the taking away of that oxygen from that heme when there's just one oxygen left to it is going to be very challenging. When you get to the second hemoglobin binding site, the second oxygen will go on a little bit easier than the first, but still a little challenging. So again, to pull off that second hemoglobin is going to be a little challenging. Right? The third one goes on a little easier, and so pulling it off is going to be a little easier. And then the fourth one is going to be super easy to get on the hemoglobin and very easy to pull off. Okay? So when you think about your hemoglobin, and we talk about blood saturation of oxygen being 98% saturated, what we're technically saying is that oxygen is touching and bound to these iron heme cores, and in any given fraction of a second, and because molecules are in constant motion, if we take a snapshot in time and we see that every 98 of these oxygen hemoglobins are being captured, stuck to each other, and then a very small percentage, 2% of the binding sites, we're going to actually, in random motion, capture maybe the oxygen and the hemoglobin in a state of microseconds of separation. So 98% saturation is the best, because what we're saying is, yes, our hemoglobins are completely saturated, and in any given free moment in time, yeah, a few of them might technically oxygen separating, but as long as it's separating and not near an oxygen demand, it's going to real quickly reattach. Okay? All right. So moving onward to the next part. Now, red blood cells, again, we add them into the system, and you can see over here the picture is showing you reticulocytes. So every day we add a few thousand red blood cells to our circulation. It comes from the trabecular bone or spongy bone areas of our long bones, known as the epiphysis. It comes from our flat bones, known, the skull bones, the hip bone, the sternum, in those trabecular spongy bone areas. All right. When the initial cell exits, it's still a reticulocyte. It still has a few more kind of last minute things to tie up, and then it will finish becoming a mature red blood cell, and its center will sink into that biconcave shape. Now, these cells will circulate through the bloodstream. Every now and then, despite whatever best efforts we do, some of these cells are going to explode. They're going to die. Okay? And when they die and they explode, everything that's inside of them is going to now become part of the outside environment. Right? So when a cell kind of explodes and dies, and again, it's going to happen because these cells are bumped and bruised into each other. They get hooked on forks in the vasculature. They get tied up or stuck, or, just things happen. They're going to, a very small percentage of them are going to blow up. So it's therefore going to allow that hemoglobin protein, carbonic anhydrase, a little bit of that glucose and pyruvate, whatever was inside the cytoplasm is now going to become part of the blood plasma, right? And again, because these cells are going to explode, that's going to mean if I look at my protein concentration, one of those other proteins I might find in blood plasma, again, very small amounts, would be hemoglobin. And if it's a plasma protein and it's a small percentage in the plasma, if I take a filter or I take a sample of that plasma at the kidney, that could lead to some hemoglobin ending up in my filtrate, and that hemoglobin could end up in my urine. Okay, so having a little hemoglobin in your urine is not the end of the world. It's kind of a natural thing because some of your cells are exploding. Now, ideally, I don't want to lose that hemoglobin because I don't want to lose those iron, heme, center core molecules. So ideally, what I want to happen is some of that plasma proteins are taken up by liver cells, and just like other plasma proteins, the hemoglobin's taken up with them, and they are recycled, made into amino acids, and sent back into the bloodstream. Some of that hemoglobin, again, and some of that cellular debris will be taken up by macrophages, which are our phagocytic cells in the spleen and the liver, and are how we get rid of some of the cell debris of red blood cells exploding. And the macrophage, when it takes in that debris, is going to, again, try to recycle some of the material. All right, so it's going to pull off the amino acids and convert proteins back to amino acids, so amino acids can go back into our bloodstream, travel to cells, and be reincorporated into other proteins. All right, now, the heme protein, most of the amino acids are salvaged, but in taking the heme and pulling out the iron core, the iron can now be sent back through binding in the liver, binding in the spleen to transferrin, can be sent back to the bone marrow and reconstituted into new heme molecules and new hemoglobin proteins. But the rest of the heme molecule that was around the iron is now at a point where I can't use it anymore. So now I need to get rid of this molecule. And the macrophages that took up these red blood cells or picked up the debris of red blood cells are in the spleen and the liver. So for the liver cells, I can quickly take that biliverdin, which is now what's left of my heme, convert it to bilirubin, send it to liver cells, and the liver cells can then take it and package it together with a few other items and put it in the gallbladder, send it to the gut as bile secretions from your gallbladder, from your liver. All right. The macrophages that are in the spleen are not able to, just give over to the liver cells because they're not in the liver, they're in the spleen. So those macrophages are going to have to take some of that bilirubin and send it to the liver, right? So those macrophages, again, are going to pull the iron off, maybe get it to transferrin, get it going to the bone marrow, the iron part, and are going to take the heme that's left, make it biliverdin, then make it bilirubin, put the bilirubin in the blood supply, and that bilirubin travels in your plasma. The bilirubin is taken up by liver cells when blood flows through the liver, and then it can be incorporated with a few other salts, a few other ions, and water, and made into bile salts and bile secretions. All right? Why is that important? Well, while we want to get rid of bilirubin, we could potentially get one more nice little useful piece out of this molecule before we dump it. And so what bile secretions and these bile salts, which include the molecule bilirubin, can do for us is when we push them into the small intestines, they can assist food that's being pushed out of the stomach into the small intestines, they can help fatty food be a little bit better suspended in the watery mixture that is now the fluid in the small intestines. And we call this bile helps emulsify fats. So it helps us suspend fats, trans fats, omega fats, fatty acids, triglycerides, small fats, steroids. It makes us cholesterol, it makes us able to emulsify them. That means suspend them a little bit easier and a little bit better in the fluid that is inside the small intestines and coming into contact with the intestinal lining, the intestinal cells that then can pull those items from the lumen of the intestines into the connective tissue, the lymphatic area, or the blood supply. All right. Now, while I want to maybe pull the fat in, I don't necessarily want to pull the bilirubin that I just secreted as part of the bile salts back in. So the main way I get rid of bilirubin is actually put it in the GI tract, and then as the bilirubin flows with the non-wanted particles of our food, it will then become part of the fecal matter. And when it gets in the large intestines and bacteria feeds off that fecal kind of last push of material before we expel it out, bacteria might be able to convert bilirubins into a few different molecules and thus contribute to our poop color, what our fecal matter color looks like. All right, so that's kind of what this slide is trying to show you. Most of our red blood cells, they live for 120 days. They go to the liver, they go to the spleen. The spleen and the liver, macrophages, so the monocytes that took up residence in the spleen and the liver, interact with these red blood cells and are able to assess that they're old by their membrane properties and by their contents of not a lot of glucose, but lots of small carbohydrate particles, 2,3-biphosphoglycerate, 1,3-biphosphoglycerate, and pyruvate, which means the cell is getting low on fuel, so the cell is about to run out of an ability to make any ATP. Okay, and then when they take up those cells, again, maximize everything we can reuse, we reuse. So the iron, the amino acids, and what we can't reuse, we convert the heme into biliverdin, which is green, so therefore biliverdin. All right, and then it turns, with a little bit of chemical reaction, turns bilirubin. Now, if for some reason you're not having your liver do a good job of pulling that bilirubin out of your blood supply, that is when bilirubin is going to increase in your plasma concentrations. And bilirubin's small, so it can start to leak out in your interstitial fluids. And when a large amount of bilirubin starts getting in your extracellular space around cells, it can start to give in your skin a more yellow tint to your skin. And that's when people start saying, oh, you have jaundice. And they're like, oh, your liver's not working. It's because it's not removing the bilirubin that's in circulation, trying to get to the liver, get uptake, get put in the gallbladder, get put in the GI tract, and expelled out. Okay? So that's where that comes from. All right? Now, red blood cell production specifically comes from a hematopoietic stem cell. Collectively, all the hematopoietic stem cells can become all the formed elements, and that's hematopoiesis. When we want to focus just on the red blood cell arm of this process, we call it erythropoiesis. So erythropoiesis is under hematopoiesis, the specific making of a hematopoietic stem cell eventually to a mature red blood cell. All right, so making a red blood cell means the hematopoietic stem cell will become what's known as a myeloid progenitor cell. The myeloid progenitor cell, under the push of certain other protein chemical signal molecules, one important one being erythropoietin from the kidneys, is going to push the number of those myeloid progenitor cells to fall into the erythropoietic pathway. Now, these cells, when they enter this pathway, are still retaining a nucleus, and are starting, because of the signal markers like EPO and some of the interleukins and cytokines and colony stimulating factors, which are just a bunch of names for a bunch of protein signal markers. They are going to start to then turn on the genes to make hemoglobin. They're going to start to turn on the genes to make the molecules on the membrane that help them as a red blood cell do their job. They're going to turn on a few genes to maybe make the glycolysis proteins, so they have an ability to break glucose into pyruvate. They're going to turn on a few genes to make, some carbonic anhydrase. Now, that's it. They don't need to continue to keep those genes going. So when they have enough of the messenger RNA at the ribosomes to make a sufficient amount of what they need, they're going to get rid of their DNA. All right. And at that point, when the red blood cell gets rid of its DNA and ejects its nucleus, it's a reticulocyte. Now, there's still a little bit of ribosomes, messenger RNA, and tRNA kind of tying up and finishing completion of all of those protein products, such as hemoglobin, carbonic anhydrase, but mostly hemoglobin. But that cell can now leave the red bone marrow as a reticulocyte and enter circulation. And then once the amount of amino acids and tRNA and ribosomes kind of are exhausted, those items will go away and the cell will be left as a biconcave mature red blood cell, jam-packed full of cytoplasm rich with hemoglobin. Okay. Now, anytime I'm going to ask a cell to go through cell division, anytime I'm going to ask a cell to do a lot of protein synthesis, I need that cell, because I'm about to unzip its DNA and I'm about to make RNA and then I'm about to make protein. I'm going to need a lot of some key regulatory factors to help make sure the DNA doesn't get damaged, and then to help regulate and control the rate of protein synthesis. And so anytime you have a growing body, a body that's undergoing a lot of cell mitosis or cell growth, you are going to need a few key molecules to help that cell be able to complete its growth process. If I want to make a lot of proteins, I need to give the cell a lot of amino acids. So bodybuilders, one of the reasons why they eat these huge protein-rich diets is because if they want to build protein, they have to have amino acids available to build protein. So they take most of their diet in the form of protein supplements and protein shakes and protein items. So therefore, once the body ingests those items in, they have a large amount of amino acids available for their cells to make into new proteins inside of the cells. So I need to make a lot of hemoglobin. I'm going to need a lot of amino acids. So if you are not getting a sufficient amount of protein in your diet, and you usually need, 0.8 grams of protein per body weight, all right, then you might be not able to make not only new cells and grow, but you might also not be able to make enough red blood cells to meet the demand that you need. Because red blood cells have a hemoglobin molecule, and the key to that hemoglobin molecule are those heme units and their iron core. If I don't have a sufficient amount of iron, I can't make hemoglobin because I don't have the heme molecules to put into those hemoglobin molecules. So in order to make enough red blood cells, I'm going to need those two basically building blocks for hemoglobin. Other things I'm going to need are the general things that help control the cellular process of DNA unzipping so I can make RNA, so transcription regulators, translation regulators, and we know that B vitamins are key to that, and folic acid is key to that. So that's part of the reason why for babies, when people are pregnant or even young babies, they sometimes supplement breast milk or supplement mom while she's pregnant with lots of B vitamins and folic acid to ensure that baby, as the cells multiply and grow, have the correct amount of regulatory and enhancing factors available. Okay. So again, when red blood cells mature, eventually we went from stem cell here. It became, because of a lot of different factors, it became a myeloid progenitor cell. All right. One daughter cell stayed and is still here, hemocytoblasts. And then under certain specific red blood cell triggers, such as EPO and other colony stimulating factors and interleukins, this blood cell now travels through that normoblast to reticulocyte and then becomes a red blood cell. Okay. So getting to this pathway is a few steps, and again, within those steps and within those cell divisions and within those cell maturation processes, I'm going to need to make a lot of protein like hemoglobin, so I'm going to need the building blocks for hemoglobin, iron, amino acids, and I'm going to need some regulatory molecules to make sure that I don't make it so fast that I make mistakes in my messenger RNA or translate it so fast I make mistakes in my protein chains, okay? All right. These red blood cells live 120 days and then get taken out. And again, the macrophages, those monocytes down here that take up residence in the liver and the spleen, they can ingest these erythrocytes or ingest the pieces of erythrocytes that may be exploded or got damaged and recycle as much as possible. So the next day's reticulocytes or developing red blood cells can have the fuel they need, the building blocks they need to eventually become efficient, effective, mature red blood cells. Okay, so again, one of the main regulations of the myeloid progenitor cell to becoming what will eventually be a reticulocyte and a red blood cell is the hormone EPO. Right? EPO is a glycoprotein. So it's protein with a little bit of a glucose or a carbohydrate on it. It's mainly made in the kidneys, and it can be made in the liver. The reason why we pick the kidneys as the main location is looking at blood supply to the organs. The kidneys have one vessel that comes into each of them. It's called the renal artery. The renal artery comes in, the renal vein comes out. Every cell that lives in the kidneys has to get oxygen from the blood that comes in the renal artery. Now, while the renal artery has a little bit of play to dilate and constrict, it really doesn't have a lot of ability to bring in more blood or bring in more red blood cells, okay? So it's a good place for our cells in the kidney to then be sensitive to, they are going to get, let's say, 200 milliliters of blood every minute. If the red blood cell count in that 250 milliliters of blood is not sufficiently carrying oxygen to those kidney cells will feel it. Right? Because there's no way to bring another 100 mils or another 200 mils into the kidneys. There's no backdoor vessel that I can collaterally dilate and pull in more blood. So that is part of the reason why the kidneys are the best place to be sensitive to red blood cell levels and oxygen carrying capacity of the blood, because no matter what happens, the kidneys get a amount of blood supply from cardiac output, and very little change in that blood happens despite whatever else is going on. Okay? And when we do not have sufficient oxygen being carried in our blood supply to our cells can become hypoxic. All right? So they can become devoid of ability to make sufficient amount of oxygen, ATP, to meet their energy demands. All right? And when we do not have enough oxygen to meet energy demands, that's a problem for cells in the kidney, because there's no way to make more oxygen appear. All right? So when these cells, in trying to do their normal functions, are not getting enough oxygen, they are going to be sensitive to the red blood cell count being low, and they are then going to trigger the release of glycoprotein erythropoietin into the bloodstream so it can travel to the bone marrow and increase the number of cells going into the common myeloid progenitor pathway towards becoming erythrocytes. All right? Now, we can map EPO, we can make it in a laboratory, so I can make shots of it and inject it into my body, and I can basically do a process of triggering red blood cell production without making my kidneys hypoxic, all right? And this is what people do when they blood dope. But, of course, we can test for this. Now, the advantage to why athletes do this is if my red blood cell count is 5.5 million per microliter and yours is 5 million, that might be the difference between my ability to run 9.5 on a 100-meter dash versus your ability to run 9.7 on the 100-meter dash. And I make the joke of, okay, in the last Olympics, who won the 100-meter dash? Actually, in the last 2 Olympics, who won the 100-meter dash? It's Usain Bolt from Jamaica. All right? Who got second? Who got third? Nobody knows. Well, I do, but I mean, I know Justin Gatlin got third last Olympics, and it was, I think, Johan Blake from Jamaica that got second. Okay. But most people know who the winner is. All right. And Hussein is a very big icon. I mean, he's like been on Saturday Night Live and other shows. He's a pretty big deal, and he's very nice. He's retired now. But you know who Hussein Bolt is. You know who the winner is. Do you know who got second place to Lance Armstrong in the 7 Tour de France's that he won? No, nobody cares because he's not number one. And so for many of these athletes, the incentive to dope is pretty high because it could mean the difference of their paycheck. Usain Bolt makes millions in endorsements. In fact, the whole reason why he didn't retire for the World Championships, that was like 3 weeks ago, was because his endorsers wanted him to do one more big event so they could have his face one more time out there. All right, and that was millions of dollars. Again, second place, they might make $100,000 in endorsements. So millions versus thousands. And so that's why a lot of these big sports that have very lucrative deals for the winners, but not much for second and third and fifth and 10th place, those athletes are pushed to consider cheating. Okay. All right. So moving on into blood typing. This again was one of our activities in labs, and I thought I went over this pretty decently with everyone, but I'm going to do it real quick one more time. So on our red blood cells, in our genes, we have proteins being made, and those proteins are or can be enzymes, can be structures like collagen and elastin, and in some cases can be membrane proteins. And in the case of blood typing, red blood cells have some proteins made based on the genetic code that can be placed on the red blood cell and can play a role in determining if that red blood cell is a self, is identified as it belongs here, or as non-self, or it doesn't belong. All right. And the genes, again, the names are terrible, but the gene that leads to one of the proteins being on a membrane of a red blood cell is known as the A gene, and it's the A antigen. Right. So the A protein, the A antigen on the membrane of red blood cells is because you have the A gene, and you would then all of your red blood cells have that gene, have that antigen, and your body knows you are self. I don't have to kill these cells because you belong. Some people don't have the A gene, they have the B gene, so they make the B protein, which on the membrane is known as the B antigen. And again, it looks a little different, but their immune system knows that the B antigen should be on every single red blood cell, and that's how they know not to kill those red blood cells. Some people have both of those antigens because they have both genes, and some people have none. So that's why they get O for devoid of either antigen. All right. So when you manipulate the process, like I said, in the Petri dish or in the kit for blood typing, I know what the A antigen looks like amino acid wise and the parts of it that are exposed to the extracellular part of the cell. And I know what the B antigen looks like. And I know then how to make a complementary protein that will, like a puzzle piece, only bind to the A antigen or only bind to the B antigen. And when they bind to those antigens, the protein, antibody, and antigen, the cell will get very heavy and kind of fall out of solution. And that's called that agglutination. Okay, so because I can make those antibodies in my rats or in my mice or in my rabbits, and then I can harness them or I can make them in the lab, I can then take blood. And what I'm doing here in this well is I have person 1, person 2, person 3, person 4. And I put person 1's blood in all 4 wells. I put person 2's blood in all 4 wells, person 3 blood in all 4 wells, person's 4 blood in all 4 wells. The next thing I'm going to do is I'm not going to do anything with the first well. So this well always represents what that blood looks like without having any other proteins added to the system. So no antibodies are added to the system. In this well, because I know what the A antigen looks like, I can then put in this well the complement to the A antigen, which is the anti-A antigen antibody. All right. Now, if that person's cells has the A antigen, that antibody will bind to it. And so my person 1 and person 3 have the A gene, and the A antigen on their membrane, so they had an agglutination event occur. All right? Person 2 and person 4 didn't. If I do a different well and I put the anti-B antigen antibody in there, again, I see that person 2 and person 3 must have that protein on its membrane because the antibody found it and bound it. All right. So in doing this, I can now say that person 1 is an A blood type, person 2 is a B blood type, person 3 is AB blood type, person 4, who never had the antibodies find the protein to bind to, must not have either of those antigens on their membrane, so they are type O. Now, completely separate gene, completely separate kind of area of DNA. I have another gene that I can manipulate and see if it's going to be a protein on the membrane of the cell, known as the Rh factor. And in the 3 people up top here, I put anti-Rh factor antigen antibody in their blood, and I found that 3 of them have the Rh factor on their membrane. So I know that not only is this person A blood type, but they are positive to Rh factor. Not only is this person B blood type, but positive to Rh factor. And not only is this person AB, but they're also positive. Now, number 4 didn't ever have the Rh factor, so I know number 4 is not Rh positive, they are Rh negative. Okay, now that is what happens in the lab because I'm going to intentionally put antibody on the blood. In a person, so this table right here is trying to show you in a person. So in my person number 1 who has blood type A, meaning they have these little green proteins on their cells, which are antigen A, I do not want them to have anti-A antibodies in their bloodstream because then that would mean their blood's aggregating and their blood cells are being killed, destroyed, and they don't have red blood cells, and that is bad. All right? So I do not want anti-A antibodies in there. Now, because they are A type and not B, if a red blood cell of a B antigen was to ever get into their blood supply, I do not want that blood cell to survive because the B gene is foreign to my genetic code, and the B antigen would highlight that that cell is a foreign cell. So what this picture is trying to show you is that if you have A blood type, theoretically, you have blood with all the A antigens on it, and circulating as part of your antibody globular protein plasma content should be some anti-B antibodies. Okay, so that's how it is for A, so you can do the opposite for B. If B is the little blue protein here, if you are B blood type, you know that your cells are supposed to have the B antigen. You know that you do not want anti-B antibodies in your system because you don't want to kill those cells. But if you ever have a cell that has the A, you want to make sure you have antibodies to that A antigen because you know that cell is not self because you do not have the A gene. Okay? If you are blood type AB, you do not want any of the antibodies against A nor the antibodies against B because you don't want those cells to be killed. And the advantage to this blood type is that you could take A blood, you could take B blood, you could accept donations from either A type or B type because your cells should have at least one, if not both of them on there. Okay. O people do not have either antigen expressed on their membrane. So they can give blood to everybody, but they can only receive blood from other O's because they would recognize any A-B antigen on the membrane as not self, and therefore their antibodies would kill them. Right? Now, the other big question that came up in lab was, what is the population of the world, you know? And most of you didn't know your blood type, but most people are actually in the world O positive, and A positive is pretty large as well. Okay, so most people in the world have either nothing but the Rh factor or the A and the Rh factor. And so I think there were 3 or 4 of us in every class, me included, that are A positive, and then usually 1 or 2 of you knew you were O, but you didn't know if you were O positive or O negative. All right, B positive is a little less abundant. O negative, again, not having the RH factor is a little bit less optimal. But O negative is really and truly the universal donor because you can give to anybody and everybody can accept your blood. All right. Again, not having the the RH factor is going to be, harder for most of the population. So again, A negative comes into play. AB positive, 3% of the world's population has that. B negative, 2%, and then AB negative actually is the most rare. All right. From your book, again, this just goes through that cartoon in a different way. If you have some blood cells, some cells that are expressing the antigen that is not self, and those antibodies find it, the aggregation, the clotting lets them fall out of solution. So we manipulate that in the lab to be able to tell what antigens are on your cells. But that process in our body will lead to degradation, breakdown, and that would be a bad thing if it happened in real life, which is part of the reason why you should know your blood type. And you should, if you ever get blood, don't know your blood type, make sure they only give you O blood so you don't run the risk of getting a bad batch of cells in your system that you recognize as foreign. Okay. All right. I've been talking for about 30, 40 minutes, I'm sure. So I'm going to stop here on this video and I'm going to pick up on the next video, part 3 of this chapter, and try to finish white blood cells, platelets, and clotting. All right. So that is where we are. We've gotten through at least half of chapter 19. Big key components are why the red blood cells are shaped the way they are, have this structure, have this content. How do we make them? What is on their membranes? Why is that important? Why do we care? And then again, overall, what's in plasma? Why? What are some advantages to that stuff being in plasma? Or what does it help us with? All those questions. Okay, so I'm stopping the video here and then I'll pick up and do part 3 so you can finish the chapter, hopefully, and have it ready before, our quiz that's coming up soon. Okay, bye everyone.